Have you ever walked past a bakery and the intense aroma of freshly baked bread has made you slow down so you can enjoy it a little bit longer? Something good is happening there. That's someplace you want to be. Now, imagine you're walking past a bank at night and you smell the same kind of bread and pastries. Weird, right? But for all you know, they're baking in there. So you slow down too. What you didn't notice were the infrared lights of the security cameras shining at you. What you also didn't know is that I modified your nervous system, so every time you are exposed to infrared light, the appropriate olfactory sensors go off and you smell bread. Sorry, there's nothing to eat here, just optogenetics. Optogenetics are a set of techniques aimed towards controlling the electric activity of neurons using pulses of light. For that, we equip the neurons with ion channels that activate in response to light of a given wavelength. These ion channels are called channel rhodopsins and are found in unicellular algae that use them to control their phototaxis. Channel rhodopsins normally respond to blue or greenish light found in their environment, but since the advent of optogenetics in the early 2000s, many new kinds have been engineered that respond to different wavelengths and that require lower light intensities. Although originally conceived as a tool for neurobiology, we have now started to employ these techniques in the fields of behavioral ecology and evolutionary biology. As sci-fi as it may sound, it is now possible to control specific neurons in a remote, non-invasive manner in freely moving animals and with a millisecond time precision, just by using pulses of light. One caveat is that for this to work, the light must incide directly on the neurons. So control without surgery is only available in animals that are sufficiently transparent, such as nematodes, fruit flies, or fish larvae. Optogenetics can work with almost any kind of neurons, but we are particularly interested in inducing sensation using sensory neurons. This allows us to alter how animals perceive their environments. For example, if you use a red light activated channel rhodopsin in Drosophila fruit flies, which do not see red light, we can provoke a sensation that the flies will likely associate with other environmental cues. So just like in the bread example, this new sensory information will be interpreted as part of the environment. For the flies, that's reality. We propose the development of this kind of virtual realities as a clean and repeatable way of delivering stimuli in behavioral experiments. Drosophila is ideal for this, as they are translucent enough, even as adults. They have a relatively simple nervous system, and there's already a lot of genetic tools available for them. Fruit flies love sugar, but in this study, they learned to avoid one of two identical sources of sugar when a bitter taste sensation is optogenetically induced on contact. So the optogenetic stimulus produces the same response as the real thing. Likewise, when optogenetic activation of olfactory receptors is paired with negative reinforcements, the flies learn to avoid the real substances those neurons detect. In our lab, we are bringing together flies that are optogenetically induced and either attractive or repellent smell, and we want to test whether this discordant perception of the environment can work as a barrier to reproduction. If these differences are able to induce assortative mating in an arena such as this without any physical barriers, we may argue that these two groups of flies are functioning as different species based only on perception and behavior. So, with optogenetics, we can influence not only the behavior of flies, but also the ecological and evolutionary consequences of that behavior. The cherry on top is that optogenetics are relatively inexpensive and very accessible, technically speaking. Adding optogenetic functionality to an existing fly lab can be cheaper than acquiring a new microscope, and not even a fancy one. So this makes for a very powerful technique that is suitable for even low resource laboratories. Therefore, we think that more people, especially young researchers, should consider delving into these techniques. And now, when you see the red light, you will want to ask your questions.